Bismillahirrahmanirrahim. Assalamu alaikum. In this video tutorial, we will be talking about restriction enzymes. So let's start. Restriction enzymes, which are also known as restriction endonucleases or molecular scissors, are enzymes produced by bacteria against invading viruses. Or we can say that these enzymes cut foreign DNA at specific sites. Restriction enzymes are actually found naturally in bacteria as a chemical weapon against invading viruses. Like if a bacterial cell is infected by viral DNA or by a virus, these enzymes prevent viral replication and save bacterial cell from that virus. So we can say that restriction enzymes restrict foreign DNA or restrict the infection of bacterial cells by viruses. These enzymes cut very precisely at specific recognition sites known as restriction sites. And as the name indicates, they cut DNA somewhere in the middle, like endonucleases. Endonucleases means that these enzymes will cut nucleic acids, DNA or RNA, somewhere in the middle, not from one end. That is why these enzymes are known as endonucleases. There is another class of enzymes known as exonucleases which cut DNA from one end. But in this video, we will be talking about endonucleases. So endonucleases, as they cut DNA in a very precise manner at specific sites, this property makes them very useful in various gene manipulative techniques like gene cloning and recombinant DNA technology. The recognition sequences for many restriction enzymes are palindromic. Palindromes or palindromic sequences are sequences that are read same forward and backward on two DNA strands, on two complementary DNA strands. Like for example, this is one DNA strand. If we read the sequence in 5 to 3 direction, it is GAATTC. On the complementary strand, the sequence is read CTTAAG in 3 to 5 direction. But if we read this sequence on complementary strand in 5 to 3 direction, this is the same, GAATTC. So if both the sequences are read in 5 to 3 direction, they are read the same, like GAATTC and GAATTC. So these kind of sequences are known as palindromic. Now let's talk about nomenclature of restriction enzymes, how various restriction enzymes are named. So each restriction enzyme is named after the bacterium from which it is isolated using a naming system. It consists of genus, species and strain. For example, if we talk about EcoRI, EcoRI is isolated from a bacterium known as E. coli or Escherichia coli. The letter E came from Escherichia, which is the genus name of the bacterium. Second and third letter CO came from coli, which is a specific name. R came from RY13, which is a specific strain of E. coli. And this Roman number I, it indicates the order of identification in that specific bacterial strain. For example, if E. coli produce one restriction enzyme, it will be named as EcoRI. If there is a second restriction enzyme isolated from E. coli, it will be named as EcoR2 and so on. If there is a fifth restriction enzyme isolated from E. coli, it will be named as EcoR5. So this is how the enzymes are named. In the same way, HindII is also named, which is isolated from Haemophilus influenza. It came from Haemophilus, which is a genus name, IN from influenza, and D came from bacterial strain, which is RD. Okay, so this is how these enzymes are named. There are various classes of restriction enzymes whose recognition sequences and cutting patterns are different from one another. Let's talk about few types of restriction enzymes. Type 1 enzymes. The recognition sites or sequences for these enzymes are not palindromic. As we talked earlier, that some enzymes have recognition sequences which are read same on the forward and backward strand. But these type 1 enzymes, they do not identify palindromic sequences. Another property is that they cut DNA far from their recognition sequences, round about 1000 base pairs away, either upstream or downstream. These enzymes are multifunctional. They have both restriction and methylase activities. That is, they can restrict or cut DNA as well as they can methylate DNA strands. That is, they can add methyl group to DNA. These enzymes need both ATP and S-adenosyl L-methionine to function properly. As these enzymes do not produce discrete DNA fragments because they cut DNA far from their recognition sequences, so they cannot produce definite DNA fragments or DNA fragments with definite lengths. These enzymes are not useful in recombinant DNA techniques. Type 2 enzymes, they cut DNA at defined position close to or within their recognition sequences. Type 2 enzymes are very specific in their cutting pattern. That is, they cleave DNA at very specific positions. That is, near the recognition sequence or within the recognition sequence. These enzymes recognize sequences that are palindromic. And these enzymes are single function enzymes, that is, they only have a restriction activity independent of methylase. They require magnesium ion to function. As type 2 enzymes is a very broad class of enzymes, they are the most common and they may recognize a sequence that is ranging from 4 base pairs to 8 base pairs in length. So a 4 base pair sequence identifying enzyme will cut DNA more frequently. The reason is that these sequences will occur about once in every 256 base pairs. Let me tell you how we calculate this number. We have 4 nucleotides, okay, adenine, guanine, cytosine and thymine, and the length of this sequence is also 4 base pairs. So we will calculate it by multiplying 4 times 4, that is 4 raised to the power 4. We will get this number. Okay, यानी हमारे पास 4 न्यूक्लियोटाइड्स हैं, एडेनिन, थाइमिन, साइटोसिन एंड गुआनिन, और इस सीक्वेंस का लेंथ भी 4 बेस पेयर्स हैं. तो एवरेज लेंथ हमारे पास डीएनए फ्रेगमेंट्स का 256 न्यूक्लियोटाइड्स आएगा अगर हम डीएनए को 4 बेस पेयर सीक्वेंस आइडेंटिफाइंग रिस्ट्रिक्शन एंजाइम से डाइजेस्ट करते हैं. ओके, अगर हम 6 बेस पेयर आइडेंटिफाइंग एंजाइम यूज करते हैं तो हमारे पास एवरेज डीएनए फ्रेगमेंट्स का लेंथ क्या आएगा, 4000 बेस पेयर्स या न्यूक्लियोटाइड्स, बिकॉज these sites will occur once in every 4096 base pairs, and this is how we will calculate this number. That is 4 raised to the power 6. तो हमारे पास ये नंबर आ जाएगा और ये एवरेज लेंथ होगा डीएनए फ्रेगमेंट्स का अगर हम 6 बेस पेयर आइडेंटिफाइंग एंजाइम से डीएनए को डाइजेस्ट करते हैं. लास्ट वन इज 8 बेस पेयर सीक्वेंसेस. Those enzymes which identify 8 base pair sequences, for example NotI, ठीक है, NotI 8 base pair sequences को आइडेंटिफाई करता है और यही वजह है कि ये डीएनए को बहुत रेयरली काटता है, about once in every 65,000 nucleotides, and that is why these enzymes are called rare cutters. Type 3 enzymes now. Type 3 enzymes also recognize recognition sequences that are not palindromic. Their recognition and cleaving sites are not same because They cut DNA about 20 to 30 base pairs away from their recognition sequences, from their restriction sites. And another class of type 2 restriction enzymes is that type 4 enzymes. Type 4 enzymes generally target modified DNA. These enzymes cut or target modified DNA like methylated DNA or hydroxymethylated DNA. Restriction enzymes may produce sticky ends or blunt ends while cutting DNA. Some restriction enzymes cut DNA asymmetrically within the sequence and leaving single-strand overhangs known as sticky ends. Okay, just like this, you can see if an enzyme like EcoRI cut DNA, it will leave single-stranded overhangs, which are known as sticky ends. And some enzymes like EcoRI, they cut DNA symmetrically right in the middle of recognition sequence, and they leave no overhangs, no single-stranded overhangs, and these kind of cuts are known as blunt ends or blunt cuts, like this. You can see the DNA is cut right in the middle of the restriction site. Now, this is how different enzymes cut DNA. If we digest DNA with an enzyme HindII, it will produce a 5' sticky end. That is, DNA is cut from here and here. So, both the strands are having an extra 4 nucleotides at 5' end, and this is known as 5' sticky end. If we cut DNA with PstI, it will leave a 3' sticky end or 3' protruding ends, like here. This stretch of 4 nucleotides, TGCA, this is known as sticky end, and this sticky end is left at 3' end. Likewise, on this DNA strand, this TGCA in this direction, in 5 to 3 direction, here in this direction, this single-stranded overhang is known as sticky end. And some enzymes like EcoRI, they digest DNA right in the center, right in the middle of recognition sequence without leaving any sticky ends. And these kinds of cuts are known as blunt cuts or blunt ends. This is another example of sticky ends produced by enzyme EcoRI. As EcoRI identify a sequence GAATTC, and it cuts in the middle of in between guanine and adenine. So it is leaving a 4 base pair overhang on both DNA strands. If we look at this strand, this is 5 end of this DNA strand and this is 3 end. Likewise, this is 5 end and this is 3 end. Okay, so if we cut DNA with EcoRI, it will leave a 5' overhang at both DNA strands, that is AATT and AATT. These kind of ends are known as sticky ends, and these enzymes are more useful in recombinant DNA technology. Now, some restriction enzymes, they identify same restriction sites and even cut DNA in a very same or similar manner. These enzymes are known as isochiasomers that cut the same manner within the same sequence, like HPA2 and MSP1. These are isochiasomers because they identify CCGG, this similar sequence, and even they produce a similar type of cut within the DNA sequence. These kind of enzymes are known as isochiasomers. There are some enzymes which are known as heterochiasomers that recognize same sequence but cut differently within that same sequence, like SMA1 and XMA1. Both of these enzymes identify a 6 base pair sequence, CCC and GGG, but both of them cut differently. Like you can see, the SMA1 produce a blunt cut and cut DNA right in the middle of the recognition sequence. But XMA1 produced 6 sticky ends and it cut differently from SMA1. So these kind of enzymes are known as heteroschizomers. So this was all about restriction enzymes. I hope you would have liked the video.